Okay, welcome to poster two on Wednesday. I would like to introduce our presenter, Xinghao Lin. He's a graduate student at Florida International University. Xinghao, do you want to advance to your poster, please?
Okay, hello everyone. So today I'm going to talk about my project. So the title is Optimizing Formia Regina sample ages from transcriptional analysis to improving forensic entomology. So at the crime scene, insects left on the bodies provide valuable resources for forensic entomologists to estimate the time of the death. And so more specifically, analysis interpreted as a post-mortem interval, which is called the PMI. So the post-mortem interval is called the PMI. To estimate the PMI, the forensic entomologist rely on two factors, development and succession. So the development means that to determine the age depending on the life cycle of the insects. And the succession, the second factor, the succession relies on the change of insect compositions on the corpses over time. And for my experiments, I mostly focus on the development of insects. And the organism I study is called the Formia regina, which is a common blowfly in US and Canada. It usually undergoes through three larva stages, which will take about more than half of life stages. What takes the more than half of life stages is the third instar stages. So now there's a big problem to determine the age of maggots. So first of all, is that so far there's no validated method to determine the true age of maggots. A second problem is there's only a little bit change of the morphology during the third instar stages, which takes, like I've been mentioned before, the third instar stage is taking more than half of life cycle. But the problem is there's not much of the morphological change. So the sizes doesn't change a lot, the weight doesn't change a lot during the third instar stages. So therefore, to finding a marker or a validated method to estimate the true age of larva is significant in forensic entomology. So one of the noticeable behavior of larva is that during the third instar stages, during that really long third instar stages, one of the noticeable behavior is that there's differences between the feeding and wandering stage. So the timing during the third instar stages, the blowfly maggots were consuming the food, and they were ceasing feeding the food before become the puber. We call it wandering stage, which the maggots were just crawling around and crawling really far away from the food resources until it finding a suitable places to become a puber. So due to this interesting behavior, I suspected there are change of gene expression during these stages. So since the gene control the behavior of the blowfly, so there must be something going on with the gene expression during these stages. So therefore, my proposed project is to examine the transcriptome profiling during the Formia regina maggots through the life stages, specifically during the feeding and wandering stages. So therefore, what will be really valuable is to identify, why I trying to do that is to identify which transcript, once I examine all the transcriptome analysis, all the profile, I can identify which transcriptome is upregulated or downregulated during the growth. But before I do any experiments, I need to optimize any sample collection and determine which timing will be really best to collect the sample, to do the RNA extraction, and to do the analysis of differential gene expression which is shown as the poster. So for the method, before I collected the sample, I reviewed the paper which been published in 2014. They doing the growth curve of Formia maggots, and of course the temperature and the amount of food will be affected on the larva growth. So I examined the references and also I did 5 different test runs to optimizing which timing will be really important. And I found during the 17 to 130 hours will be really significant to collect the sample. So what I did is like I follow the references, which will be placing the maggots in 27.5 degree and following by the circadian rhythm, which is a 12-hour light and dark periods. And so when the eggs being hatches, which have tons of maggots, I transfer those maggots to these rearing cages. So you can notice like there's a handle right there which contains a cup, and inside that has a chicken liver which will be feeding the maggots. And each handle, those chicken liver will be about 30 grams. So everything will be conditioned. The chicken liver has to be the new one, like the one I bought it from market, and it has to be the same weight. And once all the maggots have been hatched, I transfer at least 15 maggots to each of those cages. So there'll be about like 3 cages per ages. So for example, like 70 hours will be 3, 18 will be 3, and each of cages will be a 15 of maggots. So one of the most important things is I need to identify 2 different behaviors, the feeding and wandering behavior. So how I identify the feeding is that since I mentioned I did like 5 test runs, and like I noticed during the feeding behavior, those maggots will be still like crawling around those cups, but when they become the wandering behavior, those maggots will be start to falling into the sawdust. So you can see on figure 3B, which shows like the highlight one, you can see the maggots. They'll stop feeding, and they will be falling on the sawdust. And that's how I sorted between the feeding and wandering behavior. So I collected those maggots in each age cohort from 17 to 130 hours. Then I will frozen them at the -80, and then I will using them to store them and use for RNA extraction for later on experiment. And due to the COVID-19 situation, so I haven't actually got through to the sending to the RNA-seq yet. But what I'm going to do is after I collect a sample, I will extract the RNA from those maggots and send in for RNA-seq and to do differential gene expression analysis. So for the result, which I'm showing you on the poster, the figure 1 in each age cohort are weighing each individual maggots for each age cohort. So you can see all the scatter plot are represented individual maggots in each of the age. So, and sorting them between the feeding and wandering stages. So you can see during the 70 hours to 120 hours, the weight are increasing over timing until around 110 hours when the maggots start become wandering, which before they become the pupa, the weight start to decreasing. And I noticed that at 110 hours, the weight of feeding maggots are significantly different from the wandering stages of maggots. And the other important thing I need to do is I need to identify which age will be really important to identify the feeding and wandering maggots. So I also counting the fractions, population fractions of maggots. So you can see around from the 70 hours, most of them are still feeding maggots until 110 hours, where there'll be about 15% of chances of maggots they are um, half of population. They are feeding and wandering until 130 hours. The white pupils start to emerge. So overall, the preliminary data that shows that there are mixtures of the feeding and post-feeding stages for the 19 and 120 hours. So I have decided, so I actually decided to collect the sample for the 90 to 110 hours again. And also the weighting of the maggots been showing they are increasing weighting over time until it reaching to 120 hours. And finally, I would like to acknowledge both my PI, Dr. Matthew DiGiano, and my co-PI, Dr. Jeffrey Wells, and also the graduate student Amber, and also thank for Dr. Stoddard for do the 3D printing for the flying hanger, and also thanks for the fellowship supporting by the TPBS for NIH. And if you have any question, you can ask me. Thank you.
I have a question, Sheng. What's your next research areas as you move this forward?
Okay, so because right now campus is closed right now, so once the campus is reopened, what I want to do is I'm going to extract the RNA from the maggots and to do the RNA-seq, which is the RNA sequencing, the next gen sequencing, to do the counting, to examine all the transcripts from those maggots for each of the age cohort, so that eventually, once you have all the transcript profiling, you can see which gene, which transcript inside those maggots will be highly expressing at each of ages. So we can identify which gene, which transcript will be upregulated, will be downregulated. They'll be really evaluated to determine the age of maggots. And that's the purpose of my project.
Sheng, we don't have any questions yet, but we do have some new people who have entered. So if you would like, you might want to start back through your poster to generate questions from the new participants.
Okay, so so the whole purpose of my project is to finding a validated method to estimate the age of the maggots, and because to determine the life cycle of the maggots is really significant in the forensic entomologist. So due to the problems, there are two different problems. Like it's really hard to determine the life cycle of maggots. First of all, is that there's not much of the morphological change during the life cycle of the maggots, specifically in third instance stages, because the size does not change a lot during the third instance stages, and the weight does not change a lot. So one of the interesting behavior that have been founded in the life cycle of maggots during the third instance stages is that the maggots were consuming the food, which we call the feeding stages, and they will stop to ceasing consuming the food, and they're crawling really far away from the food resources, which we call the wandering stage. So due to this really interesting behavior, so I suspected there might be some gene expression changes during those two interesting behavior. So what I'm trying to do is like I will collect the maggots during those each age cohort. I was sorting them from the feeding to wandering stages. Then the next step is extracting the RNA from those maggots then will analyzes the differential gene expressions to see which transcript will be highly regulated, upregulated, or downregulated, which will be a valuable marker to identify the age, the true age of Maggots. So it's like a tracking device to see if one of the Maggots, it's whether it's 70 hours or 120 hours. So, but before I have to do any RNA extraction or to do any differential gene expression, I need to optimizing the sample, which I will collect the sample. I need to identify which timeline will be really significant to collect the sample. Also, I need to observe the behavior. So what I did is like I looking for the one comparing with one of the references, which I following the protocol based on the growth curve. They collected the forming Maggots at 27.5 hours, and I followed in the same circadian rhythm, which from 12-hour life and dark cycle. And I have this special raving container which will keep the Maggots to grow inside there. So what I did is like I preparing all the chicken liver inside this cup, which showing that in the figure. And each cup containing the same weight of the chicken liver. And I will transfer 15 new hatch Maggots into those weighing boxes. So one of really big things is I need to identify the feeding and wandering stage of Maggots. So what I've been observing so far is that the feeding Maggots will still stay inside those cups to consuming the chicken liver until when they reach into the wandering stages, those Maggots will be fall on the sawdust, which shown on the figure 3b as highlighted in red. You can see the Maggots that will be falling onto sawdust. And I'm collecting, so I'm sorting them between feeding and wandering stages into at each of age cohort from 17 to 130 hours. And what the preliminary data showing on the figure 1, which I weighing the each individual Maggots during the development, that shows that during the 17 to 120 hours, the weight of the Maggot will be increasing. Now, at 110 hours, the weight of feeding Maggots are significantly different from the wandering stages. And the other thing I do is that I counting the Maggots between the feeding and wandering stages and also the white pupil, which showing that the 110 hours, that shows about half of population of feeding and wandering stages, which is really important to collect a sample. So this will be really helpful to identify the transitions of feeding and wandering stages. And until about 113 hours, which shows the white pupil start to emerge. So for the conclusions, the preliminary data shows about the suggestion mixture of feeding on wandering stages from 19 to about 120 hours, which will be in the next step. I would like to collect a sample again and then to do the RNA extraction. So the other thing is that the Maggots weight shows the increasing weight over time until 120 hours. And that also at 110 hours, the wander feeding and wander wing, they are significant different. And finally, I would like to acknowledge my PI, Dr. Matthew DeGenevo, and my co-PI, Dr. Jeffrey Wells, and grad student Amber, and also Dr. Stoddard for providing those 3D printer of the handle. And also, I would like to acknowledge the fellowship. Thanks for fellowship supporting by TBBS from NIH. And thanks, everyone. So if any questions, you can ask me.
Yes, and we have some new people in, so if you have questions, if you review the poster, please put those in chat. I do have a question about the 3D printing. Is that unique? Is that something you developed for this research? The holder, the sample cup?
It's actually really, yeah, it's actually Dr. Starter, he designed it. So we have a kind of conversation, discussion. Well, will be really good thing to, first of all, to identify the feeding and wandering makers. That's a really important thing to point out. And also, we want to design a device that the makers won't escape in a container. So we've been discussing, and Dr. Starter, he been really creative to designing this handle, those 3D printing handle, to put the food inside there.
One follow-on question: have you thought about capturing this on time-lapse video to look at the development of the maggots?
Oh yes, actually, I actually captured a video before one time, and I did thought about to doing that, and because you can actually observing the behavior of makers over time, but this still little bit trouble because um they still have a little bit trouble. I have a problems I have encountered is that first of all is that the makers following by 12-hour light and dark period, the circadian rhythm. And sometimes like if you turn off the light, like if you cannot see the behavior of maggots very well. And you might miss the timing when the wandering behavior of maggots fall on the sawdust, which is one of the problems we encounter. And also I trying to optimize the graphic of the camera as well.
Yeah, it's interesting though that could be very useful by using some new imaging techniques to do measurements as well as enhance your research when you're doing the next gen sequencing. I think I asked you that question before. Maybe some of the folks didn't hear that. What's your next goal once the campus reopens?
Okay, so first of all, I found it like 19 between 19 and 110 hours is really important. So I don't need to collect the sample those again, but I would like to recollect the sample for 19 to 110 hours. And the method, there'll be a little bit change because during this experiment, I just directed frozen them at the -80, which I will be optimizing by just frozen those bakers, just kill them at the liquid nitrogen right away, so that the metabolism will be stopped at the right timing. And then I will do the RNA extraction right afterwards and sending to the company to do the RNA sequencing so that you can counting the transcript inside those makers during these life stages. Then you can identify which transcript will be really, which one will be really useful, which transcript will be really useful. There'll be really good marker to identify like the age, the true age of the makers.
Okay, I have a question for you.
Yeah.
The question is, do you have a fly colony in the lab where you can get the eggs from? How big
Oh
is that
yes.
colony? How big is that colony?
How big? Um, I do remember it's actually I never counted the 5 people, but we do have a colony of the fly inside the lab. And if I just visually to see the fly inside there, at least about hundreds of fly inside the cages.
Okay, thank you. That was from one of the participants. Seeing how we have a new presenter or a new participant that's come in, if you want to go over briefly your poster and see if they have any questions, that would be great.
Okay, so one of the most important job for the forensic entomologist is to estimate the time of the death, which is known as the PNI, the post-mortem interval. And one of the factors that forensic entomologists to estimate the age is based on the life cycle of the insects. And one of the most common insects maybe you can find on the corpses is the blowfly. So the tool I'm being studied is the organism I study is the Formia regina. It's pretty common profile in US and also in Canada. And so one of the problems to determine the true age of maggot is that, first of all, there's not much of morphological change during the life stages of maggots, specifically on the third instar stages of larva. It's the last instar stages of the maggots before they become a pupa. So when I say no morphological change, is that the size does not change a lot, the weight does not change a lot. So it's really hard to determine the true age of maggots at each age cohort. So the other problem is that there's no validated method to determine the true age of maggots. So one of the noticeable behavior of maggots is that when the maggots during the third instar stages, the maggots were eating the food without consuming the food, which we're calling a feeding stages, until before they become the pupa. There's a timing when they start to ceasing eating the food, which those maggots will be wandering, they will be crawling far away from the food resources, and we call it wandering stages. So due to this two interesting behavior, those transition of this behavior, that might implying that there must be some gene controlling those behavior. So maybe the gene expression will be really valuable resources to identify one of the particular marker that will be really useful to identify the true age of the maggots. So the most so my objective of my experiment is to examine the transcriptal profile of maggots during the life cycle, specifically from the feeding to wandering stages. So before I have to do any RNA experiments, to do any RNA extraction or do any differential gene expression, I need to optimize the sample of maggots. So first of all, I need to observing the behavior of maggots and also need to identify which of the age cohort I need to collect the sample. So of course, I only interested from the feeding and wandering. So I'm not going to collect all the sample for the entire life cycle. So first of all, I follow the comparing with protocol, which the person did a growth curve of Formia maggots, and are following the same temperature, which the researcher did, and also following the same of the circadian rhythm, which is a 12-hour life and dark period. So I have this special rearing container, which shows on figure 3, that I will put the maggots for, those maggots will be growing for the life cycle inside those boxes. So, and also what I did is that once all the maggots has been hatched from the eggs, I transfer 15 maggots into those special boxes, these rearing boxes. And you can see there's a carpet containing they are containing a chicken liver. So all the weight of the chicken liver has been conditioned. So those weights will be the same, about 30 grams. And each box is containing 15 maggots. And one of the biggest things I need to sorting out is the feeding and wandering behavior. So what I've been observing so far is that during the feeding of the maggots, they will just keep eating the chicken liver inside those cups. And they'll just coin around those cups. And when they stop to ceasing eating those foods, those maggots will be falling on the sawdust. So you can see on the figure 3B, which show on the highlighted, those maggots will be fall on the sawdust.
they'll be sorting as the wandering stages or Maggots. So I collected the Maggots at each cohort for 17 to 130 hours, and at each cohort I counting the numbers, the fractions of the population for feeding and wandering, and also white pupil, and also I weight individual Maggots at each cohort. So in the preliminary data, it shows that the weight of Maggots are increasing over time until 110 hours, and the weight start to decreasing. And at age 110 hours, the weight of feeding Maggots, they are significantly different from the wandering Maggots. And on the figure 2, that shows that the feeding of the Maggots, they are about 100%. And you can see at 110 hours, the feeding and wandering stages of Maggot, it's about 50% of the Maggots until about 130 hours, the white pupil are start to emerge. So in conclusion, those preliminary data that shows that for based on the fraction of populations, there's a mixture from the 90 to 110 hours. And also about the other thing is that the weight shows that for 17 to 110 hours, the weight of Maggots, they are increasing until about 130 hours. Those weights start to decreasing before they becoming the pupa. And finally, I would like to acknowledge of my 2 PI, Dr. Matthew DiGennaro and Dr. Wells. And also, I would like to acknowledge your grad students, Amber, and also thanks for Dr. Starter for providing those handle to put the put those chicken liver inside there. And also, I would like to acknowledge the fellowship support by the TPPS for NIH. And thank for everyone. And if any questions, you can ask me.
We do have a question. The question is, so every 10 hours, you remove all 15 maggots, weigh them individually, and then return them to the food bowl.
No, actually, sorry, I forgot to mention, I frozen them at -80. They'll be using for the RNA extraction later on.
So you don't take them out and weigh them every 10 hours?
I'm sorry, can you repeat the question?
So the question was, so every 10 hours, you remove all 15 maggots, weigh them individually, and then return them, or you freeze them after that?
Yeah, I freeze them afterward.
Any additional questions, please use chat. I did have one more question for you, Xinghao. Is chicken liver the best food supply for these maggots? Is that what you found?
So lots of paper has been actually the food resources. The liver is a part of the important nutrition resources for the maggots, and it's been using a lot to rearing those maggots. But there are some paper that they're still using the liver, but just from different animal, like from some that I found they're using the beef, and lots of people also using the chicken as well. So we just using the common one, and also I also noticed some people even using the heart of the animals to rearing those maggots.
Yeah, I just is interested in seeing that was the food source of choice.
Yeah.
So where do you get your chicken livers from?
I just bought it. I just bought it from supermarket right away.
Okay. So were they frozen or fresh?
It's actually fresh because I need to. It's the best to condition all the food resources. So everything has to be fresh. Like the liver has to be fresh. So once I would like to start the experiment, I need to go to the supermarket and just buy the chicken liver right away.
I noticed that you had the food cups 3D printed. Were the other containers just commercially purchased, or did you 3D print the whole device?
Actually, we just 3D printing the whole device.
So the external clear container as well as the food cup.
No, those boxes, it's actually just those boxes are not 3D printing. Only those plastic handle, they are 3D printing. And boxes just bought it from market.
Okay, got it. Will you be doing the next gen sequencing yourself, or are you going to use a service for that?
No, I would like to send in a sample, the RNA sample to the company to do the RNA sequencing. And so I will receiving the data, then to do the analysis.
All right, we're not seeing any new participants come in. I'll give everyone a few minutes to get back to the main session. Maybe grab yourself a glass of water or a cup of coffee. I'd like to thank Tsinghao. I see Mason's in parentheses there. Sorry, Tsinghao, I was calling you that instead of Mason. But we really appreciate the research that you're doing, and thank you so much.
Thank you.